Hi everyone, welcome to this video on B1 cells. So I'm just going to talk through animal and plant cells, the organelles, and then a bit of magnification. So let's get started then. So an organelle is just something that is found inside a cell. So it's the smaller parts of the cell, for example, the nucleus or mitochondria. It's just those little parts of the cell which you need to be able to label, you need to be able to recognise them on a diagram and also explain their function, function meaning job. So let's start with an animal cell. So I've got this image here, let me just fold it in half, and we're going to label the organelles and also I'm going to go through the function. So here is an animal cell. You can recognise that it's an animal cell just by the shape. So it's usually circular or circular-ish, and the first organelle that you need to know is the nucleus. So this one here, it's usually larger than everything else, just a circular shape. That is the nucleus, and the nucleus basically controls the activities of the cell. So for 1 mark in the exam, when it asks you what is the function of the nucleus, you just say controls the activities of the cell. Around the cell, so you can see here this thicker line, that is known as the cell membrane. So the cell membrane it's like a gate, it just controls what goes in and out, controls what goes in and out of the cell. Because you don't want everything flooding in or everything flooding out. There has to be some sort of control in order to protect the cell. Here we have, so in the entire cell, we have a jelly-like substance known as cytoplasm. So on my diagram, it's white, the same as the paper, but it might be a different colour, and it's just all of this in the background. It's a jelly-like substance known as cytoplasm, where chemical reactions take place. Okay, then we have these tiny dots. They're usually really small in the diagram, so that the tiniest dots you can recognise as ribosomes. So ribosomes basically make proteins. It's where protein synthesis happens. That just means where proteins are made. So protein synthesis happens here. So there's one thing missing from this diagram actually, so I'll just draw it in here, and that's mitochondria. So mitochondria have lots and lots of folds like this, and that's how you're able to recognise them. These folds here increase increase the surface area of the mitochondria, because this is where energy is released. So mitochondria is the site of respiration, so it's where respiration takes place and therefore where energy is released, and that shape just increases the surface area, so more respiration can happen and more energy is therefore released. Okay, so that is your animal cell. The other one is a plant cell. So if I just flip over and just wipe the board. This is a plant cell, and you can tell it's a plant cell because it's more of a rectangular shape, and it's also got a thick cell wall around it, which an animal cell does not have. Usually exam questions will ask, you know, what are the differences between an animal cell and a plant cell, and there's 3 main differences. So I'll just go through the differences first. First one being this cell wall. So can you see this thicker rim around the plant cell? That is the cell wall. And the cell wall is there for support and protection. So don't get confused with the cell membrane, because it might ask you to label the cell membrane as well. The cell membrane is the thinner one that runs around the inside. So just this thin line here running all the way around. The cell wall is the outer one, which is thicker. So cell wall is one difference that an animal cell doesn't have. The other thing is a vacuole. So a vacuole, you can see here, it's really large, so much bigger than the nucleus. So always go for the really large one, being the vacuole. And this contains cell sap. So cell sap is just sugars and salts for the plant cell. Sugars and salts for the plant. So cell wall, vacuole, and then the other thing, the other difference is chloroplasts. So chloroplasts are the reason why leaves are green. So these shapes here, usually on your diagram, if you get a coloured diagram, they will be green because they contain a pigment called chlorophyll. And the chlorophyll basically absorbs sunlight. So absorbs sunlight for photosynthesis, because without sunlight, plants can't photosynthesize. So for photosynthesis. So there are similarities between animal cells and plant cells. So like I said, you've still got the cell membrane, you've still got a nucleus, you've still got ribosomes, you should have mitochondria as well, so I'll just quickly draw that. Mitochondria. You've still got cytoplasm, the jelly-like substance running all the way around. So there's only 3 main differences to remember. So the other thing I will go through is that animal and plant cells are known as eukaryotic cells. So eukaryotic, if you were asked in the exam to name a eukaryotic cell, always say animal or plant cell. So those are your only 2 options really, so just go for those 2. And eukaryotic just meaning there are membrane bound organelles inside that cell, whereas prokaryote or prokaryotic cells are bacteria cells. So it could just be a 1 marker in the exam, just be sure to quote those. So going through a bacteria cell then, so I'll just draw this one. Bacteria cells, again, are usually maybe rectangular, oval, not as round as an animal cell. They have a cell wall, so there's some similarities and some differences between animal and plant cells and bacteria cells. So the bacteria cell has a cell wall, again, for protection, for support. It's got a cell membrane, the inner one. Got cytoplasm, that jelly-like substance where chemical reactions happen, but they do not have a nucleus, so no nucleus. Their DNA, bacterial DNA, is either in these hoop shapes known as plasmids, so that's not a nucleus, that's just a hoop of DNA, or you find them as these squiggles known as free DNA, so like a squiggly shape, and it's known as free DNA. Bacteria cells sometimes also have a tail known as a flagellum, and the tail is basically there to help it move, help it swim, just like a sperm cell has a tail to help it move faster. The bacteria cell also has a flagellum. So that's all you really need to know about cells. So let's go on to magnification now. So there is an equation to know, and I will go through the triangle with you. So magnification, it's basically about how much you can zoom in to something, especially cells. We're always trying to zoom into cells to have a look at the nucleus, have a look at the organelles. One other definition you need to know is resolution when it comes to microscopes. So microscopes have magnification and they have resolution. Resolution is how clear the image is. So you don't really want a blurry image when you're looking down the microscope. So resolution is the official definition, the ability to distinguish between 2 points. So I would learn this definition word for word, the ability to distinguish between 2 points, and all it means is how clear is the image. So a light microscope is the one you tend to use at school, so you've probably done this practical with a lamp and a microscope and looked down and maybe looked at like a leaf cell or an onion cell. So light microscopes have poor magnification and a lower resolution than an electron microscope. So an electron microscope, they tend to be really expensive. You wouldn't really have these in school. They have a greater magnification, they're more powerful, greater magnification and higher resolution. So always remember that the electron microscope is better than the light one in all ways, the magnification, resolution and power. So the equation then to work at magnification, and there, especially in a paper 1, there is usually a magnification calculation. So magnification equals image size divided by actual size. I'll just explain what these mean. So image size is the image you get on your piece of paper. So in the question there might be a little box and there might be a picture of like, I don't know, like a leaf. So that is your image size because that is the size of the image you are given. The actual size is the size that it is in real life. So in most cases the actual size tends to be bigger because you've zoomed in, because you've magnified it and you're looking at organelles or whatever the question asks you to look at. So all you have to do is take your ruler. Let's say it's talking about the width of this leaf. You take your ruler and you just measure the width of the ruler like that. Usually it's in millimeters, but just be careful. See what the question asks you. If it's saying to do it in centimeters, do it in centimeters. Otherwise, I would always just stick with millimeters. And then the actual size will be given to you in the question. It might say the width of the leaf is really 20 millimeters, something like that. And then you just plug it into your equation. Let's say I measure this and it's 2 millimeters. So therefore my magnification is image size 2 divided by actual size, which is 20. And they're both in millimeters, so therefore I don't need to convert anything. So that's one main thing to look out for. Students often lose marks because they forget to convert the units. So actually I'll quickly just draw the triangle for you and then I'll go through converting units. So for anybody who prefers to learn the triangle, image size goes at the top there, magnification on the side, and then actual size here. And then if you want to work out magnification, you cover magnification and it'll be image size divided by actual. If you want to work out image size, you cover the eye and you do magnification times actual. And then if you want to work out actual size, it's image size divided by magnification. Right, so conversions then. So we started with millimeters. If I just put millimeters there in the box. In order to go from millimeters to micrometers, let me do it like this, micrometers, and that symbol is, it's like a U with a stick at the front. To get to micrometers, you have to times by 1,000. So millimeters times by 1,000 puts the units into micrometers. Again, if you want to go from micrometers to nanometers, you times by 1,000. So you're getting more each time. So if I had 50 millimeters, that converted into micrometers is 50,000. And then again, 3,